Welcome from the San Antonio Breast Cancer Symposium. My name is Dr. Julie Nangia, and I am a breast oncologist from Baylor College of Medicine in Houston, Texas. And with us we have
Andrew Tutt. I'm a breast cancer oncologist at the Institute of Cancer Research in King's College London.
And today we're going to talk about one of the abstracts that was presented in the first general session here. And this was an update on the OlympiA trial. And the OlympiA trial was a phase 3 randomized trial in high-risk germline BRCA carriers to look and see if there was benefit from adjuvant olaparib. So Dr. Tutt, maybe you can tell us a little bit about the original clinical trial design and the results, and then we can go into the updates.
Thank you so much. So yes, OlympiA was designed, as you say, in the context of high-risk HER2-negative breast cancer in patients who'd had chemotherapy, either in the neoadjuvant or adjuvant setting. And it was designed to test the sort of therapeutic principle of targeting a defect in homologous recombination in patients who'd got germline mutations in BRCA1 and BRCA2. The therapeutic principle has nothing to do with whether or not someone's HER2-negative breast cancer expresses the estrogen receptor or not, but we separately stratified those patients because, of course, their treatment algorithms are different, and to some extent their risk profiles by stage were different. And so it tested 12 months of olaparib therapy after the standard of care chemotherapy, surgery, and other local therapies as in a placebo-controlled double-blind design. We're presenting the data here at San Antonio on this occasion as a third pre-planned analysis driven by the fact that this is now 10 years from first patient in to the study, and with a median follow-up now of just over 6 years across the study. So an additional 2.6 years from the last presentation of the trial.
Yeah, and just as a reminder, everybody, I'm sure you all know, but this is a study that led to the FDA approval of this drug for adjuvant use. And we'd love to hear what are the updates showing and what more do we know?
So for the primary endpoint, we now show that there is the rate for IDFS at 6 years is 9.4% difference from the placebo arm. For distant disease-free survival, again, an increasing difference, now 7.8% absolute difference between the olaparib and the placebo arm. And the overall survival benefit that was shown to be statistically significant at the second interim analysis has again strengthened with a rate difference of 4.4% at 6 years. We also show that there is no evidence of any difference in subgroups pre-specified for analysis in the trial. So importantly, the benefits of olaparib extend to both the triple negative and the ER positive HER2 negative population of patients. We could define no subgroup where the effect of olaparib was not present, and all of the confidence intervals cross the same point estimate as the main trial effect in the whole population.
Yeah, and I thought there were some other interesting tidbits that came out of the presentation. One, it was that they didn't really see any difference in future pregnancies, which I think is very reassuring to women. And the other was looking at the risk for secondary cancers. Maybe you can comment on that a little bit.
Yes, so you're referring to our analysis of new primary malignancies, where we continue to show that with longer follow-up, there are fewer new primary malignancies in the olaparib arm than in the placebo arm. And we did set hypotheses around the fact that BRCA, so targeting BRCA defective cancers that tend to occur in this population, breast and ovarian cancer, we might actually reduce the incidence of these BRCA mutation-associated second malignancies with a year of olaparib. Now, numerically, we do indeed see those differences. This is quite a complicated analysis because it is obviously affected by risk-reducing surgery and the timing of that risk-reducing surgery. But overall, the risk-reducing surgery rate for risk-reducing surgery or ovarian risk-reducing surgery was the same between the two arms. and yet there is a numerical difference in contralateral breast cancer events and in new ovarian or fallopian tube events reduced in the olaparib arm. So that is promising and interesting. We'll continue to follow this up with more analyses that are able to take into account the timing of risk-reducing surgery, which is important to consider.
And then the last question I have is a real-world question. So a lot of people are wondering, well, now that we have olaparib, how do we use immunotherapy in that triple-negative subgroup? And do you do both together? Do you sequence them? Do one or the other? What is your advice on that? I know we don't have the answer. We wouldn't be wondering this.
No. So OlympiA was conducted in a period when the Keynote 522 study had not reported and this wasn't approved therapy. My own view on this is that OlympiA applies to patients who've got residual disease after neoadjuvant chemotherapy in the triple-negative setting, and we know that patients who've got residual disease after Keynote 522 therapy still have significant risk of distant recurrence and overall survival events. And there is no reason to believe that there is cross-resistance between immunotherapy and olaparib. These are drugs which have been combined safely in a number of other trials. And so I think there is it's perfectly acceptable to consider giving both the adjuvant immunotherapy phase and adjuvant olaparib in those patients. I would not suggest waiting until after completion of immunotherapy to begin olaparib, because we know that the curves separate very early after surgery and in follow-up in these patients, and that is where we're seeing the prevention of recurrence by the use of 12 months of olaparib.
Thank you so much, and thank you everyone for joining us on the updates from the OlympiA trial.
Thank you.